Assalamu alaikum and good morning, guys. Uh, we are going to continue from where we ended up last time, and we have discussed some basic concept about lipids, where we studied about different classifications and different biological functions of the lipids. So today we are going to start as one of the very important biological function of the lipids as the structural component. And when we say that the role of the lipids in the structure, the most common thing that came into our mind are the membranes. So you can see the picture of a membrane in front of you. I won't go in much of the detail, but just a couple of points in this diagram. You can see that this membrane is a lipid bilayer structure. So it has got two lipid layers. The way the lipids are arranged is very important. You can see that these dots here on the top and on the below surface, these are the polar sides of the lipid which are facing the water environment, because outside the cell and inside the cell you have got water environment. While this yellow portion which you can see sandwiched in between these polar heads is actually the non-polar part of the lipid, what called hydrocarbons. So the way the lipids have been arranged is they are exposed on the either side with their polar groups facing water molecules or water environment. And in the middle, we have the hydrocarbon non-polar portion, which is not facing the water environment. So that's the basic assembly of a lipid when it comes to the cell membrane. So going in further details, when we talk about lipids as an example of structural composition, the most common example that came into our mind is, as I said, is other membrane lipids. And that's how I showed you a membrane lipid in the picture. Now, within the membrane, there are different varieties of the lipids present. And you can see in this flow chart, there are certain varieties of the lipids. You can see phospholipids, glycolipids, then archibacterial lipids, and sterols. Now, first to start with the phospholipid. Phospholipid is a type of the lipid we call the complex lipids. जैसे मैंने पहले आपको बताया था कि when the lipids are attached with some additional group. So in the phospholipids, it's phosphate group attached with the lipids, and this is one type of a complex lipid. Similarly, glycolipids have got the carbohydrate moiety attached with a lipid, and there are certain archibacterial lipids which are definitely with reference to the microorganisms, the type of the lipids present in the membranes and the cell walls, etc. And then we have a specific type of the lipid, what we call the sterols, which are also very important and integral part of the membrane lipids. And when we talk about the sterol, the most common animal sterol that is present in the membrane is cholesterol. Now, the phospholipid can be further classified as glycerophospholipids and sphingophospholipids or sphingolipids. Again, this is a further classification where we go in the details of the structure to see that in phospholipids, when the alcohol molecule is glycerol, it's called the glycerophospholipids. And in case if the glycerol is substituted by sphingosine, which is another alcohol, that's called as sphingolipids. Then in the glycolipids, it's carbohydrate with the lipids. They can be further classified as sphingolipids and galactolipide, or in short, sulfolipids, and then so on and so forth. So we go in further detail to see this is a molecule of a glycerol phosphate, where you can see that this is a 3 carbon glycerol, जो कि हमने पहले भी discuss किया हुआ है, if you remember. In terms of the tricell glycerol, हमने पहले भी discuss किया था. So glycerol is a 3 carbon alcohol, and you can see that one of the hydroxyl group, we have a phosphate group attached here, and that is what we call the glycerol phosphate. Technically, the name of this molecule is L-glycerol 3-phosphate, because on the carbon number 3, we have a phosphate group. Now, this molecule is the backbone of all glycerophospholipids. Now, this is the general structure of our glycerophospholipid. and you can see the same molecule of cholesterol, and we have a phosphate group here, and then we have the rest of the two hydroxyl group. We have ester linkage, just like tricyl glycerol, and that is being shown here, two fatty acids, which could be saturated or unsaturated. Now, in this case, we have one saturated fatty acid, which is palmitic acid, and one unsaturated fatty acid. You can see a double bond here. That is the oleic acid. So the type of fatty acid that connects to the L-glycerol phosphate are very specific for different organisms. So that means in a different organism, there are a specific type of, the alkyl phosphate connecting with the different fatty acids, and they have different functions within the cell. Here are some of the glycerophospholipids commonly present in the body: phosphatidic acid, phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine, and phosphatidylglycerol. The most common difference in all of these phospholipids or the glycerophospholipids derivatives, you can see, is the type of the alcohol, like ethanolamine, choline, serine, glycerol. These are different types of the alcohols, and the precursor molecule for all of them is phosphatidic acid, which is being shown here. Now, going to another type of the lipids, which are called the galactolipids, and these are the lipids which are probably the most abundant of the membrane lipids, and they primarily and predominantly exist in the membranes of the chloroplast, thylakoid, etc. For example, 70 to 80% of the vascular plant membranes are made up of these galactolipids. And now again, you can see that we have a glycerol molecule here. We have fatty acids attached, and we have a sugar moiety in place of the phosphate, in contrast to the previous example. So these are the examples of galactolipids. Now you have one sugar moiety, or you may have more than one sugar moiety. So that gives to the mono or the di concept. So these are the galactolipids. Primarily, 70 to 80% of the membrane of the vascular plants have that structural lipids in them. Then we have the sulfolipids. Again, the name is pretty much clear. They have a sulfate group substitute. Unlike the phosphate group, this sulfate group is not directly substituted at this hydroxyl position. Rather, we have glycerol, we have two fatty acids, and we have one of the carbon attached with the sugar moiety, and the sulfate group is attached on the sugar moiety. So that's what we call the sulfolipids. Sulfolipids, they also have a group, as I said, sulfur group or a sulfonated group, which is attached, to a diacylglycerol, and this particular bond is called the glycosidic linkage. You know about the carbohydrates, and this kind of lipids are also predominantly present in the chloroplast. Then we have certain archaeobacterial lipids, which I mentioned earlier on. These archaeobacteria are the one that live with some very extreme environmental conditions. These are certain microorganisms which live in certain extreme conditions, like very high temperature or very low pH, high ionic strength, like the difference of ionic balance on either side. And for these reasons, because of certain extreme conditions of like temperature and the pH and other factors, they need membranes which can resist these conditions. And these archaeobacteria would have specific types of the lipids which have long chain carbon and branched hydrocarbon, and that gives it more strength to face certain extreme conditions, as we mentioned. Uh, Another type of the lipid which was being mentioned earlier on is the sphingolipid, and the sphingolipids are actually the derivatives of the the molecule of sphingosine. And you can see here this purple molecule. This purple molecule is the molecule of sphingosine, and you have got one of the fatty acid substituted here, and this X represent any other moiety, which could be anything else. But this is the basic structure of sphingolipid, where you have the molecule of sphingosine, and we have got fatty acids substituted here. So sphingolipids are the derivatives of sphingosine. They are having one molecule of sphingosine, which is an alcohol like glycerol, one molecule of long chain fatty acid, and a polar head group. So they have one polar head and two non-polar tails. Why two non-polar tails? Because one is this hydrocarbon chain, one is sphingosine itself, which is non-polar, while this X is what we call the polar part of the sphingosine. So you can see that the common thing is, like in the sulfolipids, we have sulfate group, which is the polar group. In phospholipids, we have the phosphate group, which is the polar group. And in sphingolipids, we have the polar group here, just giving these molecules bipolar nature. Like they have polar group and they do have the non-polar groups. Sphingolipids can be further classified into different types like sphingomyelins, glycosphingolipids, and gangliosides. Now we'll go in the detail, but just to make sure, giving you some idea, sphingomyelins and gangliosides are predominantly present in the nervous tissues. So this is the structure of sphingomyelin, and you can see that there are two molecules in this category. The basic molecule of sphingomyelin or sphingolipid is ceramide, which is the backbone, and the sphingomyelin is a group where you have got X molecules substituted by phosphocholine. If you remember this last here, that's what I mentioned. This X is going to be substituted. So when this X is substituted by phosphocholine, then this will become the formula. So sphingomyelins, they contain phosphocholine here, or phosphoethanolamine, which is another molecule, as their polar head group, and they are classified as phospholipids as well. So it's pretty much clear that the ceramide is the basic structure, where you've got X molecule, and this X is substituted by either phosphocholine, or it is substituted by phosphoethanolamine. So they've got a phosphate group in that, and that's why classified as phospholipids as well. For sphingomyelin and phosphatidylcholine, they are very important aspect of the structural part of different membranes and protective membranes. And here's a picture showing you the three-dimensional structure of these two molecules. There's nothing important in this, rather than just showing the hydrocarbon chain, the alcohol molecule, and if there is any charge group, like here the polar group is phosphate, here we have the polar group phosphate. So this is the 3D structure. Now sphingomyelins are very, very important lipids, and they are present in plasma membrane of most of the animal cells, and very especially, as I mentioned, in the nervous tissues. They are especially in the myelin, which is a protective covering of neuronal cells. You might have studied about the myelin sheath. Myelin sheath is a membrane, a protective membrane that insulates the neuronal cells, specifically the axons. So sphingomyelin is the structural component of that myelin sheath structure, which insulate the axons of some of the neurons. That's why I said that sphingomyelins are important lipids structure in the nervous tissues. Then we have the glycosphingolipids, and the name itself reflecting that you have sphingolipids, and now you have got an add-on to that. and the add-on is a sugar moiety. So when a sugar moiety is added to the sphingolipids, it will become glycosphingolipids. So glycosphingolipids include different categories. They have different categories of the molecule, and glycosphingolipids include cerebrosides, which is one example, and globoside, which is another example of glycosphingolipids. Primarily, they occur in the outer face of the plasma membrane, and the reason is that they have polar group in them, so that's why they face the outer part of the membrane where they have water environment. Cerebroside, which is one example of glycosphingolipids, they have a single sugar linked with the ceramide molecule. So you can see here, this is the baseline ceramide molecule, which is backbone, and then you have a glucose molecule uh, that is attached via glycosidic linkage to the ceramide, resulting in the formation of cerebroside. While the globoside has got two or more sugars. So here is the molecule of the globoside where you have got two or even more than two sugar moieties attached. They have no charge generally at the pH 7, and that's why we classify them as neutral glycolipids. So at a normal pH 7, they are not charged, so they are being classified as neutral molecules without having a charge. Then we have another example of structural lipids, which are called gangliosides. They are also a very important molecule when it comes to the nervous tissues. Gangliosides, they have the sugar moiety, but that sugar moiety is an oligosaccharide. So if you go back here, you have got either one sugar moiety, which is being called as cerebroside. You have got two or more than two sugar moiety, that is being called as globoside. And when you have more than two, or rather I would say an oligosaccharide moiety attached with a ceramide, that results in the formation of gangliosides. And they have a attachment through the N-glycosidic linkage. This one you can see here. So uh, their head groups, or the charge group, or the polar group, whatever you call them, they have more residues of N-acetyl neuraminic acid, which is shortly being called as salic acid in a common term. And this is the structure of a salic acid here. Now, as I said, that gangliosides also have got some important role when it comes to the membrane lipids. And here is one of the very, very important role of gangliosides, because gangliosides are also the part of the membrane lipids, and they predominantly play the role when it comes to the determination of the blood group. So you are aware of the fact that the blood groups are being determined. There are specific surface antigen on the membrane of the red blood cells, which help us to identify the blood groups. So it's actually the gangliosides which have predominantly the role in determining of the blood group. So you can see here, this is the ceramide, and we have sphingosine, and we have the fatty acid, and then this portion is the oligosaccharide I was talking about. This is the oligosaccharide. So you can see that in these three examples, the oligosaccharides have some differences. The rest of the molecule is the same. So that oligosaccharide attached with these three have got some differences, based on which we have got O antigen, A antigen, and B antigen, which is the blood group O, A, and B. And we've got the both A and B antigen, that's the blood group A, B, right? So this is the important role of the ganglioside in the body. Now, this was all about different varieties and different types of the phospholipids, and we tried to be focused on different types with a specific information about their structure, what sort of group they have, and a little bit of information about their function or biological role when it comes to the structural component. Now, going forward, these phospholipids, they can be degraded by certain enzymes within the body, and the group of the enzymes that are required to hydrolyze phospholipids are being classified as phospholipases. The phospholipids are degraded by specific lysosomal enzymes, which are being defined as phospholipases. So you can see here that this is a molecule of a phospholipid, and you can see phospholipase A1, phospholipase A2, phospholipase C, and phospholipase D. Now, these are the four examples of the phospholipases, and you can see that these four enzymes are working at a specific site. They're very specific in their role. Like this phospholipase A1 is going to specifically break this particular bond here, and then phospholipase A2 is going to specifically act on this bond, and phospholipase C is going to hydrolyze this phosphate bond, and then phospholipase D. So these phospholipases are very specific enzyme when it comes to their target bond, and they hydrolyze phospholipids. Definitely when these enzymes act, that's going to hydrolyze the phospholipid molecule into its constituent molecules. Now, let's discuss some clinical conditions related to the phospholipids. When there is hydrolysis, as we mentioned in the previous slide, then there will be a breakdown of the phospholipids. And when these phospholipid molecules are broken down, they might result in certain derivatives. And these derivatives, they may have a certain role, but if they are being accumulated somewhere, they might result in certain clinical disorders as well. So I'm just going to discuss few of the examples here. Like if you look at this molecule starting from the ceramide, so remember that ceramide was the backbone molecule, the precursor molecule, and then from the ceramide we can have different varieties of the molecules. So in this case, you can see that uh, you know uh, this enzyme galactosidase will result in the hydrolysis of this molecule from there, and then we have a couple of other enzymes resulting in different derivative formations, and these derivatives are being summarized here as well. So if these membrane lipid derivatives they undergo abnormal accumulation, they might result, as I said, in certain clinical conditions. And some of the main symptoms of clinical conditions associated with accumulation of these derivative molecules include mental retardation, paralysis, blindness, or even early death in certain cases. Now, the last of the type was sterol. So sterols are, specific category of the lipid molecules which contain this basic nucleus. Now, this ring structure, rather, I would say it's a structure which is formed by four rings. So we have three six-carbon ring, or six-membered, I would say, not exactly carbon, and then there's a five-membered ring. So we have three six-membered hexagonal and one five-member, which is pentagonal ring. All of these four are fused together, they are connected together, and this collective molecule is actually being referred to as the nucleus of all sterol molecules. And when it comes to sterol, the most common sterol that came into our mind with reference to animal, I would say, is the cholesterol. But yeah, there are other sterol molecules as well, but the most common is the cholesterol. So this four ring fused structure, this is being referred to as the nucleus of the sterol molecules. And the name is very important. It's a little bit difficult, but you have to learn for that. This ring is called cyclopentano perhydrophenanthine ring. This four rings fused together. That's the basic nucleus. They are structural lipids. Sterols are also structural lipids, and they are the integral part of the membrane of most of the eukaryotic cell. And cholesterol is the major sterol in animal tissue. So this is the basic nucleus of all esterol molecules. And you can see in the cholesterol, this is the cholesterol structure, and you can see that this nucleus is there, this one. So the difference comes that all sterols would have this nucleus, but they have difference of this hydrocarbon chain that results in different varieties of the sterol molecules. So we are talking about the cholesterol, which is the most important. Cholesterol is, under the category of dry lipids. If you remember the classification, simple lipids, complex lipids, and then there was dry lipids. So cholesterol is actually in the category of dry lipids. It's widely distributed in animals and in humans. Very, very essential part of the cell membrane. It has got a very specific function. You know that cell membrane, you have studied that it's semi-permeable, and you have studied that cell membrane is a fluid membrane. It has got a fluid mosaic model, and that fluidity of the cell membrane is primarily because of the cholesterol. So cholesterol is very important molecule when it comes to the fluid nature of the cell membrane, which allows certain molecules to pass through the membrane. Similarly, cholesterol is also important when it comes to vitamin D, certain hormones, bile acids. These are all molecules which are synthesized from the cholesterol. Bile acids, they are important for the digestion process along with the bile salts, and they are having a role in the absorption of the fats and fat soluble molecules from the diet. Most of the cholesterol synthesizing site is liver. Uh, Definitely, it's an important molecule, but if there is an increase in the cholesterol molecule, that would result in certain issues as well. If you have increased cholesterol, it has the tendency to accumulate within the arteries, causing atherosclerosis or the blockade of the arteries, and that leads to coronary heart diseases. Unlike the cholesterol, unsaturated fats, uh, they have reduced level of cholesterol, while the saturated fats would have higher ratio of the cholesterol in them. Now, for the cholesterol, we classify them in three categories of the molecules based on their densities. And that's a very important parameter you might have heard about when we do the blood testing of our total lipids. We came to know about these molecules, and these molecules have got different indicators to indicate a good type of the fats or the bad type of the fats in our body. So we have LDL, HDL, and VLDL. LDL is the low density lipoprotein, and high density lipoprotein, and very low density lipoprotein. So as I said, that they have certain clinical markers as well. You can see here that low density lipoproteins are the one that transports cholesterol from the site of its manufacturing, that is liver, to via the blood to the tissue. That's particularly what we call the bad cholesterol, because if it's more, that means more of the cholesterol will get a chance to accumulate within the arteries. High density lipoprotein, they are the one that transport cholesterol from the blood back to the liver, where it is going to be metabolized. That's being called as the good cholesterol. Now, why we are calling it the bad or the good cholesterol? Because of the fact that how the cholesterol has got a chance to accumulate or will be taken back to the liver. So, high density or the low density, this density actually is the parameter which determines the role of that particular molecule. So you can see here, this clearly indicates that when you have increased level of LDL, that means you have increased level of the cholesterol. And when you have increased level of the cholesterol, you've got a high risk of heart attack. And just, reciprocate, when you have high level of HDL, you have a low level of cholesterol and a low level of or low risk of the heart diseases. So this is why I call this a clinical parameter to understand. Then bile acids are also the derivative from cholesterol, as we mentioned, and this is the structure of the bile acid, and you can see this four ring structure, what I call the nucleus of the sterol. You can see that here. So that shows that yeah, it's made from cholesterol, that is a sterol. So bile acids are actually the polar compounds. They are the polar derivatives of cholesterol, and because of their polar nature, you can see here this polarity is here. Because of their polar nature, they act as a detergent, and when the lipid molecules come in the gut, in the intestine, we need certain molecules to emulsify the lipids, as you know, and that's how it acts as a detergent to emulsify the lipids to help the enzyme to digest the lipids. And that's how when the bile acids or the bile salts are there, because of their polar nature, they emulsify the fats, the dietary fats specifically, and then the lipases, will act on the lipids and will result in the hydrolysis. Finally, a couple of information regarding fatty acids. Although इस presentation के शुरू में हमने fatty acids के बारे में पढ़ लिया था, but there is one classification that I want to share with you. Fatty acids के बारे में एक तो आपने पढ़ा कि they can be either saturated or unsaturated, and you know the criteria. Saturated fatty acids, where you have got all the four valencies of the carbon satisfied by a single covalent bond, and unsaturated fatty acids, where you've got multiple, double bond or multiple unsaturation. There's another classification of the fatty
देखो
acids
जवाब
based
दो
on.
सही से।
There is another
सही
classification.
वाले दो, पहले जवाब दो सही वाले।
Guys, you have been advised many times to keep yourself mute. I don't know what's wrong. In every class, there is one incident of someone accidentally unmute. And I have got the names here, who used to do that. In the next class, I'll, take you out of the class because that's going to disturb the class for everyone. So make sure that you should be equipped enough to know how to mute and unmute. So now the fatty acids, they can be classified as essential and non-essential fatty acids. Now, what's the criteria? The criteria is the essential fatty acids are those which your body cannot synthesize. That means you need those fatty acids coming through your diet. It's just like essential amino acids, if you remember. So these fatty acids, they must be present in the diet because your body cannot synthesize them. So when it comes to essential fatty acids, it's linoleic acid, linolenic acid, and arachidonic acid. These essential fatty acids, they can be synthesized. They can use for synthesis of other molecules in your body, like prostaglandins, which are hormone-like structure, leukotrienes, prostacyclin, thromboxane. These are all the modulator molecules which help in different processes within the cell, in the regulation of the blood clotting, inflammation, reactions or infections, etc., etc. So essential fatty acids, if there is a deficiency in your body, as I said, you cannot synthesize them. That's why if the fatty acids, essential fatty acids are deficient, they result in the deficiency of these molecules which I mentioned. And that's how it results in a poor growth and a couple of other clinical conditions, immune response, etc. So the best dietary sources of the essential fatty acids are vegetable oil and other sources. Then we have trans fatty acids. I'm not going in much detail, but if you remember, I told you when we studied about the isomerism that there are two configurations, cis and trans. So that's what we are talking about, the trans fatty acids. These trans fatty acids, they exist in a very small amount. Naturally occurring fatty acids, जैसे मैंने पहले बताया था, they are having a cis configuration. So there are very few amount of trans fatty acids in the natural food. Trans fatty acids, they lower the HDL level and raises the total blood cholesterol, as we discussed earlier on. They also raise plasma concentration of the lipoprotein. Trans fatty acids are formed when vegetable oils are hydrogenated during the formation of the margarine. The vegetable oils are converted to vegetable ghee and then the margarine by the process of the hydrogenation. So that process actually results in formation of more of the trans fatty acids. And that's why we say that vegetable ghee or margarine, these are unhealthy product when it comes to, because they have trans fatty acids in them. So these are some key points. about what we have studied. Lipids are structural elements like glycerophospholipids, sphingolipids, galactolipids, sulfolipids, archaebacteria, and other sterols. Glycerophospholipids are derivatives of the phosphatidic acid. Common glycerophospholipids are phosphatidylethanolamine and phosphatidylcholine. And then galactolipids are composed of diacylglycerol with a galactose residue. Chloroplast membrane are remarkably rich in these type of the molecules. And then we have sphingolipids contain sphingosine alcohol and one long chain fatty acid, have three subclasses. Then sterol you have studied, they have got four fused ring, which is the nucleus and a hydroxyl group. And cholesterol is the major sterol in the animals. So with this we have completed lipids, and with this, if you have any question, you can raise your hand, and I'll or you can write down in the chat box. uh, I will inshallah update this lecture once it is available, and we'll share the link with you guys. If you have any question, please raise your hand now. Okay, so if you don't have any question, we are going to finish at this stage. Just to remind you that this Monday, sorry, this Wednesday, we would be having our first lab at the time, which is as per schedule, inshallah. And I will share the recording with you guys, and inshallah see you for the lab on Wednesday. Thank you very much. Khuda Hafiz.